Hi everyone, welcome to my channel Srinivasa Biology Tutorials. For this section to discuss NCERT Class 12, the chapter is Biotechnology Principles and Process. Part 7, obtain the foreign gene product. Insert the piece of alien DNA into the cloning vector. It is transferred into as a bacterial cell or any plant or animal. So here, anyone completely the alien piece of the DNA, it is they're introduced into as a plant or animal. The alien DNA get act as a multiplied. What happened? The one DNA is get produced as a multiple copies. Am I right? One DNA get forms as a multiple copies. In the most of the recombinant technologies, the ultimate aim to produce as an undesirable protein. So mostly which purpose we are doing this total process, we get developed to as in a multiple copies. Am I right? It is mostly in a antibiotic resistant gene we are completely developing. Let us these foregoing genes get expressed the appropriated conditions. This expression of the foreign genes, the host cell involved under understanding many the technical details. So in this total, the foreign genes we can introduce into as a host cell. What happen to get producing as a more there, what kind of the genes we require easily to get develops. After having cloning the genes of insert having as in their optimized conditions including as expression of the target protein. So this total process based on target proteins which one have considered as a producing and as a large scale. So the number of copies, how much quantity you require, you can easily to get develops. Now you can think any reason why they have need for as a large scale production. If in case we require as a large scale, how do they process? If the protein encoding the gene expressed as a heterogeneous host is called as a recombinant protein. So simply that recombinant DNA we get attached with as in where heterologous host cell, that is called as a completely recombinant protein. Next, this cell as in the harboring cloned genes interested by as in that growing in a small scale the laboratory. So in this interest, it is only the growing as well in a they're completely in a laboratory. Okay. Next, this culture may be used for in a extracting the dissolved protein and purifying using as in different separation techniques. So this total process when we are using as in a different separation techniques based on we get develops. Next, let's see, these cells can be their multiplied continuous culture system where in used as in a medium they are drained out from outside of the fresh medium is added into as in another maintained the cells as in a physiological moist active long the exponential phase. So in this total process the physiologically active long phases we are developed as in this total materials. Next, after this type of culturing method, see this type of culturing method to produce as in a large biomass leading. In this culture method producing as a large biomass leading higher yielding desirable proteins. So they get produces a number of copies at a time. Let the small volume culture cannot be yield upgradable the quantities of products. Let's see here. The produce a large quantities development of bioreactors. So the small we get developed as in that small laboratories, but large quantity we require so we developed is where we get used as in bioreactors. Whereas in a large volume around as in 100 to 1000 liters of the culture can be the process was required. So minimum 100 to 1000 liters we required. We get completely get using as in a bioreactors was required. See, this bioreactor mostly. Let's see here. These bioreactors. Thus bioreactor can be the through the vessels which can be raw materials of biological converted into specific products. So biological here mostly the raw materials biological conversion into as their specific products. This is here we are using as what individual enzymes. Here individual enzymes also to get help to as to get production of the multiple copies of the DNAs etc. Let's using as a microbial plants and animal or human cells. So in this process we are Not only one single purpose we are using this one. We are using as what? Microbial plants as well as animals and then human cells also. Okay, let us. Here, bioreactors is provides as an optimal conditions for achieving the any desirable products, the providing the optimal growth condition like temperature, pH, their substrate, salt, vitamins, oxygen. This all completely to get maintained. Then only you get as in what? You get as in a proper product. The most common used bioreactor in a steering type, which is shown the give diagram here. So it is in a the steering type of bioreactor. It is to get producing as what? That completely the 100 to 1000 liters of their materials as get produced. This is in a mortar, foam beaker, flatted blades, and culture growth and sterile airs. This completely to get maintained as in a they're completely in a bioreactor. Okay, next last one here. A steering tank reactor usually the cylindrical or within a curved base to facilitate mixing of in a reactor the reactor content. So it is a cylindrical shape. That cylindrical shape it is the complete that mixing of in all the contents. Next, the sterile facilitates even the mixing the oxygen availability through as in a bioreactor. So when we are adding this what we are completely the giving as in a their oxygen availability. Here they giving as what? Here completely has any oxygen availability. So when the oxygen availability is present, then what happen to automatically to get produced as the alternately air can be bubbled through as in a bioreactor. If look as in a figure closely, you will see in that bioreactor see aggregated their agitator system. So mostly in the agitator system to get producing as what? In a completely to get oxygen delivery system and foam control system and temperature control system, pH control system and sampling pots, their small volume of culture withdrawal periodically. So these all things we are seeing this the diagram representation. So here completely to get the given in the diagram. Okay, na? This is. Next, last, that is downstream process. The last one is what? Downstream process. What is the downstream process? After the completion of any biosynthetic stage, the product also they are subjected through as any the series of process before it is ready to marketing as in a finish. So finished product. So after to develop as in this total product, how do they get separated? How do they get purified? Without with any the purified, we can't allow it as in a market. Am I right? Let's see here. This process separation. This process includes the separation and purification, which are collectively refers to as a downstream process. So separation and purification of the DNA is called as what? In completely downstream process. This product has to be they're formulated with as in a stable the preservative. So mostly the all preservative based on in this total DNA we are get they are separated. Such formulation as we undergo through the clinical trials as causes drugs. So the number of clinical trials also they are required and strict quality control testing and each product also they are required. So everything the quality control or testing of each product be there completely to required. Now the downstream process is quality control testing vary from product to product. So this all is complete then only these products will get transferred their transferred into as any uh, marketing and companies whatever. Okay, this is a incomplete downstream process. Remaining we discuss in next class. Thank you for watching. Please share, subscribe and like. Thank you.